Okay, you can see the start page. Okay, thank you. Because all I need is a nod of the head. Okay. This will be in the way, so let me click it off if I can. I want to go back to the start of the week and quickly go through a couple of things. The website. The website is not a part of Canvas. I had it put together years and years ago, updating it with each class. And now for you, it's updated to summer 2021, okay, with your ticket number and such. But you were asking me the other day about reading assignments. Okay, so I went as I promised and looked at reading assignments last night, and now here are your reading assignments. But the only three you have so far are the introductory chapter, that's one, the joints chapter, that's nine, and those were both assigned to you for the first time on Monday, 6/7. Well, I changed now my wording for the date that they're due. These are due at P1. Now, you may have to run to the syllabus to see what date P1 is, or you can just remember that we have a practicum every 2 weeks on Thursday. So next Thursday, these chapters 1 and 9 are due before the practicum. They won't be acceptable for credit after the practicum. Okay. And now I've assigned, I know you're going to say, wasn't it chapter 2? If you've started chapter 2, go ahead and continue. I would prefer now to uh, shake some clarity into my head. I should have given you chapter 6. Chapter 6 is ossification, and that makes sense. We just talked about it. That's being assigned today, and that's due. Now, what's due for P1? 3 chapters. This one, this one, this one. Or if there's a few of you that have already started the cell chapter, chapter 2, you may turn in the 3 for 1, 9, and 2. I need 3 of them from you for next Thursday. Somebody may be gung-ho and say, well, I don't want to do the cell later on. I've already got it started and finished, but I'd like to do ossification because that's more relevant right now. You can turn in 4. I'll keep track of it for you, and then you won't have to turn in the other one for P2. Okay. I outlined in the class uh, parts of the chapter that don't need to be read and emphasize those that do for chapters 1 and chapters 9. But I'm not going to do it, even though I said yesterday that I would do it. But I realized that in the ossification chapter, the whole chapter short is necessary. So I would like you to do the entire chapter 6. Okay. Uh, for chapter 2, if you're working on it, you can email me and say what parts should I do on chapter 2. I'm not going to go over that with the whole class because the whole class will not be doing it. But I'll have a sheet ready for you, and I can send it to you if you're doing chapter 2 as to what to outline.
And
Okay.
Charlie,
Yes.
and you said, I think it was in the email or you said it in person, that if we're doing chapter 6 this week, if we haven't done chapter 2 yet, that we'd be doing chapter 2 next week.
No, I didn't say that. Uh, that may be what you thought I was implying. Okay.
Okay.
Uh, I will. I said I need only 3 chapters from you before P1. It could
Got it.
be the first 3, or it can be the first 2 and chapter 2. But I need 3 chapters. Okay, and then where's the other chapter going to be assigned? Well, obviously it's going to be assigned next week, but or thereabout, but it won't be due until P2. I have the feeling you're still a little confused, or somebody is out there.
No, I
Just
got it.
well, I didn't mean that you're still confused. I just have the feeling once in a while when I'm confusing somebody. So they should speak up and tell me they don't understand quite what I said. You got it? 3 chapters outlined. Now I've got some chapter outlines in. I haven't been able to open Laura. I haven't been able to open yours. I don't have the program. I don't deal with Apple. I think I told you that. So please, if you can convert that to a Word or a PDF file, that's best all around. And also, uh, I otherwise have not looked at the outlines. I'll tell you that honestly. I usually do that on Friday, okay, and then get back to you only if they're not satisfactory. And satisfactory really is a relative term. Satisfactory is an outline that helps you on your future tests. And if it doesn't help you, then it's, or I think it won't help you, it's unsatisfactory and we've got to deal with it and bring it up to snuff. Okay. But don't expect that I will come back with a decision right now. But the decision in 90% of the cases is your outlines are fine. In 10% of the cases, it's either your outline is too short, this outline was done at the last minute and is of no value to you. You okay, something like that. There'll be a comment so that you'll know uh, why, if I don't think it's satisfactory, why I feel that way and what I think you can do to improve it for yourself.
And
Okay.
Charlie,
Yes.
it'd be fine if we sent it in like 3 different PDFs, right?
You can send it in, but I'd prefer it not be in 3 emails with the PDF attachments.
Okay.
Attach all 3 of the PDFs to the
To the
same
same
email
email. Okay.
is nicer. Okay, I can keep track of things a lot better that way. But the best thing is, of course, get them sent in. Okay, and I'll deal with it no matter how I have to. Okay.
Carly, I have a question.
Yes.
For the entire chapter, does it matter how long the outline is? Or do
It
you only
usually.
want it to be like 1 to 2 pages still?
Oh no, I didn't say the number of pages. Although I may have indicated that I often get 1 and 2 page outlines. I've had outlines as short as a third of a page. Well, I'll tell you what, that's not good enough because there's more information in every one of these chapters than that. I just want you to outline the important material. If it takes more than 2 pages to do that, then you should do more than 2 pages.
Okay, thank you.
Don't be afraid or ashamed, if that's the word, of turning in just a couple of pages. On the other hand, that's not necessarily bad. The outline is to shorten, to condense the material so that you have a sort of a study guide to refer to when you're getting ready for your lecture test without going back and rereading the chapter. That takes way too long to do that. Let me go back here. I want to point out a couple of things to you. Uh, Again, you know this, the web links in Unit 1. You're going to be on your own for 5 days. I was at 4 days in the summer here. You're going to be alone over the weekend. And you want to see bones, you can Google bones. You can get so many different things from different schools. but this first link is on the bone box is very, very handy. I've shown it to you, so I'm not going to open it up again. Okay, but it's very handy. Also, we have been doing the skull. We did the vertebral column yesterday. These are just lists. These lists don't have the figure locations as they do on the module. Okay. We're going to be doing the upper appendicular skeleton today, and finally the lower appendicular skeleton on Monday. I may forego lecture on Monday to do the lower appendicular skeleton and then move right in to the actual bones from the bone box, so we have plenty of time to look at them. That would be the rest of Monday. That would be Tuesday. That would be Wednesday. These are days of review, okay, to learn this material. But just because they're sitting out there and you know you're going to have some review time, you don't know what else is going to demand your time. The dog scratching at the door, wanting its food, your kids, if you have them, whatever. Something could come up. Take advantage of the weekend. I don't want to ruin your weekend, of course not. Now that things are maybe starting opening to open up, maybe there's something that you can do safely and enjoy. But don't let whole days go by without taking a look at the material. Okay? The knee, we'll get into that on Monday. Don't worry about that. The idea of x-rays, there are a few x-rays on the practical exam where you identify structures from x-rays. Well, why not? If you're going to go into the health field, are you going to see bones or are you going to see x-rays? Most often you're going to see x-rays. You're going to see a bone if it's a compound fracture, and that's going to be it. Or if you're in surgery, you might see a bone. So we need to take a look at x-rays. But believe me and trust me, this will not complicate matters for you at all. People have no additional trouble moving from our pictures or images to the x-rays. So I'll do that on Monday. I want you to look at the tissue slides. There's only 5, I believe. Okay, we'll start with the top one. Sort of a fuzzy picture, but it's obvious, it should be anyway, what this is. This is compact bone that we saw yesterday. These are osteons, each one of these. The haversian canal is dead center. The lacuna, like little bugs going around in a circle. The lamella are not clear, but you know where they are, but they're not clear and I couldn't ask it. This little guy here is a start of a haversian canal. It's not running in the plane that this piece of bone was cut. It's running downward or upward. There's no way to tell, and disappearing. But it really does continue on. Be able to recognize compact bone. Be able to recognize cancellous or spongy bone. This is more of a model over here of what it would look like. And this is the real thing. Has lots and lots of lacuna, large depressions. We've talked about cartilage. The articular cartilage at the end of the bone is hyaline cartilage. Here it is here. Sometimes when you're first learning a bone, or a tissue, I should say, you have to think of something that it looks like and reminds you of. What does this remind you of?
Eyeballs.
Eyeballs, okay. If that works for you, keep it.
Reminds me of a beehive.
A beehive, okay.
Looks like white blood cells, kind of.
Reminds me of a coral reef.
Yeah, and a coral reef, bunch of little islands. No, also look like a bunch of people on their surfboard waiting for a wave. No, this
I
guy's
can see that.
okay. Anyhow, it also looks like molten plastic here. It always does. Now, never rely on the color of a slide because it can be stained any color. Okay, look at the structure that you're seeing. We're going to see this plenty of times before you get asked it next Thursday. This is elastic cartilage. Okay, and now we haven't come across elastic cartilage yet in the course, but elastic means something can be bent out of shape and then flip right back into shape again. So you've been dealing with this for a while. If you put a mask on around with the loops around your ear, you bend your ear and your ear pops back up. We know that. It does with or without a mask. And this is the cartilage that's doing it right here. It's more dense. Elastic cartilage. Your epiglottis, if you know what that is yet. If you don't, well, later in the course you will. The epiglottis and the ear are the two major locations of elastic cartilage. Now, you have to have a textbook or somebody to explain it to you. You won't always have me to explain it. So a textbook. This is a rod in this particular instance. Okay, a cylinder. And it's been cut. So it looks flat. It looks like maybe a marble pathway. Okay. And on the side here, this is connective tissue or and smooth muscle. Okay, we don't have the cartilage in our ear, for instance, right on the surface. It's buried in tissue. Tendon is this last one. Tendon is known as dense regular connective tissue. Dense because, well, it is dense. Okay, regular because the marbling tends to go parallel. Now, there's a little bit of waviness that you can see in here, and that's not in you when the tissue's in you. This is because of the cutting of the tendon, which is hard, and you're using a knife called a microtome machine. And when it cuts it with a waver like this. You never really or rarely see one cut perfectly straight. This is tendon. Okay, I'll get that off of there. And that's all I wanted to show you on the website. Every time you're studying the material on Canvas, I still would appreciate you go back to the website for Unit 1. When we get to it, Unit 2 and 3 and 4. Okay, now I'll get rid of that. Okay, your week 1 assignment. I told you I would do some changes on that, and I did that last night. Okay, the assignment objective is to outline chapters 1 and 9. Okay, and then it says if you've already started chapter 2, you can continue with it. You don't have to continue with it. You can put it aside and finish it off in the next section. Okay, you may wish to switch to chapter 6. Okay. Now, when are these due? All of them now, I've changed the date from the suggested date of Sunday night coming up. I said to you that they're always due before the next practicum. The next practicum is June 17th on Thursday. and that'll be at 1 o'clock. So they need to be in by 1 o'clock for credit. Otherwise, it goes down as that you missed the assignment. And if you miss the assignment, oh God, what happens then? What happens if you don't get your the assignment in?
You don't take late assignments.
I don't grade the assignments.
You don't get extra credit?
That's it. You don't get the points for the previous weeks of attendance. You want them. I know you do. Okay. I'm not trying to coerce you into getting these assignments in, because if I were going to do that, I wouldn't repeat to you. You do not need extra credit to get an A in this class. It certainly, though, helps. And if you want it to help, then you got to do it. You got to attend, and you got to do the assignment. And the assignment in this case is 3 chapters, 169 or 16 and 2, 19 and 2. Your choice. Okay, I think I've said enough on that, so that if you don't understand it, email me, please, and I'll be happy to talk to you. Please finish the meet and greet if you haven't put your 2 cents in there. It's interesting to know how many people are already in the medical field, how many are planning on going into the medical field, who the top gamer is in the group. I used to play video games, the stand-up arcade. I used to spend $300-400 a month playing those games until I mastered them, and then I'd put a quarter in and I'd stay all day.
Which game was it? Or was it more than one?
Oh, more than one, but Ms. Pac-Man. I think I have the Orange County record for Ms. Pac-Man. Okay. Here's the recording for last night. Okay, so we want to see it. There it is when you need it. One thing good about the recording, you can just pull the bar across and skip to the part of the recording that you need. So if you were there for the first part, you can just move to the last. I almost feel apologetic to do these two things here. But as I said, levels of organization and the structure of the cell, something that we all did in our first biology classes, is forgotten by many students. It's the most frequently missed set of questions. And should it be? Of course not. Okay. And if we review it, it won't be. We don't need that to happen. So I do have to go over it. I'll try to go over it rapidly. If you think it's too rapid, or maybe it's the first time for you, I don't know how you got in this class if it's your first time, but you can slow me down easily enough. We'll start with levels of organization. This won't take us long at all. Cells are the lowest level of organization that we start with. Somebody else, a physicist, will start with subatomic particles, etc. But we start with the cell. It's considered to be the smallest living entity, and it's capable of quite a few things, including reproduction. Okay. We'll be going into the cell in the section right after this in a few minutes. Cells that work together to perform a common function are called tissues. Tissue, composed of similar cells that perform a common function. The word common doesn't mean familiar or commonplace. Common means the same function. They share, they're synergistic. So we go from cells to the next higher level, which is the tissue. The tissues can be one of four major types. These definitely need to be remembered. We'll be going over them throughout the whole course. Epithelial tissue. Epithelial tissue is the cut tissue of the body that covers the body or lines the body. So you feel the lining of your mouth inside, that's epithelial tissue. If you swallow a marble, so it won't be digestible, and it goes all the way through you, it's touching epithelial tissue all the way through. Your blood vessels are lined with epithelial tissues. Epitheliums or epithelia cover or line. It's as simple as that and as complex as that. So remember, it can be outside, it can be inside the body. Then we have nervous tissue. We'll leave that for future study, a lot of future study. Muscle tissue and the connective tissues of the body. We're looking at connective tissue right now. Bone is a connective tissue, the blood within it, and the cartilage that is on it or makes up the synchondroses of the epiphyseal plate. These are all connective tissues. A little diagram or cut and paste of four types of tissue then. Epithelial might look like this. Here's one called connective tissue. This is probably a relar connective tissue. Nothing that you have to know yet. Muscle tissue. Well, this is skeletal muscle and nervous tissue. And they're just showing here one neuron, another neuron. Okay. I don't think these pictures are of any benefit. Somebody just plucked them in there. Probably me. Okay. When tissues come together to perform a common function or functions, they're called organs. If I ask people to call off the names of tissues, I get very few answers. When I ask people to call off the names of organs, it's nonstop. Stomach, pancreas, heart, skin, liver, brain. What's another organ?
Lungs,
Lungs, sure.
Kidney.
Kidneys.
skin,
Skin is the largest, oh, it's here. Okay. Skin is the largest organ in the body or on the body. I have so many people, and you know, I don't do trick questions, but this isn't a trick question. At least it shouldn't be. I ask people in a multiple choice question, which of the following is not a tissue? And I put in some, but there's only one that's not a tissue, of course. And that answer is skin. It's an organ. And people miss it over and over and over again, and then they kick themselves when they see the correct answer because they know it, but they just can't bring it up front. Okay. Of these six, you know, and maybe the ones that you added, which are the two organs that utilize the greatest amount of energy day and night?
heart and brain,
You got one of them. The brain is one of them. It's not the heart, though.
I would say heart and stomach, maybe.
Oh, none of those are correct.
It's the brain and the liver.
pancreas.
It's the brain and the liver. Okay. Oh, hey, what you just skipped ahead to the next sentence, didn't you? The
No,
brain
I
and the,
can't read. Yeah.
it's right there. Okay, the brain and the liver utilize most of the energy, especially when you're resting, compared to the other organs of the body. Okay. When organs come together to perform common functions such as digestion, well, then you have an organ system, and the system then would be the digestive system. Here are the organ systems. And if you recall yesterday or the day before, when we went through chapter 1, I pointed out 12 pictures, dark pictures, and said, don't bother with them at this time. Those were pictures of the organ systems. And we're going to go over every one of these organ systems that you see here, okay, individually at a later time. Okay. Not later than 7 weeks from now, since we're going to have our first week under our belt. And then when all the organ systems come together to function, you have the organism. Those are the levels of our hierarchy of organization. Now go backwards, start at the largest: organism, organ system, organs, tissues, and cells. And now we look at the cell itself. Review of cell structure. We haven't talked about cell structure, so it's not our review. This is a review of what you learned whenever you learned it in another college class, high school. I got it in junior high school. Okay, I'm going to limit this to the things that you have to know. Here's the cell. The membrane around it is the plasma membrane, plasma lemma, or cell membrane. Oh, great. 3 different ways of saying it. There'll only be one presented on the multiple choice test, but you should recognize the correct answer from those. Okay, this is the plasma lemma. Very, very important, not only because it contains the cell, keeps it inside, but it determines what can come into the cell and what can leave the cell. It's also, of course, going to grow and expand as the cell reproduces. The cell membrane. The cytoplasm. Everything outside of the nucleus, between the nuclear membrane and the cell membrane. This is all cytoplasm. That includes the organelles that are inside that we haven't named yet. This is all the cytoplasm. There's a term called cytosol. I want to see if I gave it to you, so let me look down. Cytosol. Can you write it for? Do you have a pencil today? Or if I need to, I'll print it up. Cyto, C-Y-T-O-S-O-L. The cytosol is the fluid part of the cytoplasm. Think of a swimming pool. You've got a pool party scheduled. People are coming over. You hired somebody to clean the pool and get all the leaves and stuff out. The pool has nothing in it but water. That pool represents the cell, and the water represents the cytosol. Now I should throw a big ball in the pool to represent the nucleus as well. But the cytosol, the water, has nothing in it. Now the people start arriving and acting foolish and jumping into the pool. They're jumping into the cytosol and becoming part of the cytoplasm. Now it's cytoplasm. So all of the inclusions between the nucleus and the nuclear membrane make up the cytoplasm. The nucleoplasm is the material inside of the nucleus. Okay. Now I could expound upon that, and boy, can't tell you the number of lectures, crazy lectures I had at UCI, a crazy school, by the way. I was in the first graduating class at UCI. I was in the first class that ever took a class at UCI. But that's another story, a little bit later. The nucleoplasm is the area within the nucleus. Chromatin. When I first learned to draw a picture of the cell, you could hear all the other students with their pencils tap, 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 tapping their picture. Well, that's because that was the chromatin, the pelleted looking material inside of the nucleus. What chromatin is, it's dispersed genetic material. That means it's dispersed DNA and dispersed messenger RNA, okay, and it's dispersed. What does dispersed mean?
Spread out.
Spread out. Good. Think of a plastic rope. Have you ever been down to the harbor? They tie up ships, yachts, and cabin cruisers, everything with plastic ropes. They're usually yellow or blue. You've all seen these plastic ropes, okay? And they're made of many, many, many fibers, all twisted together tightly. Now think of that as a chromosome, a length of twisted plastic rope, maybe 3 feet long, and it's really tight. Think of that as a chromosome, okay? When a cell is dividing, you can see the chromosomes under the microscope. Okay? And the chromosomes are tightly wound, which protects them, which protects their genes, the sites on them that carry the genetic information. It stops the chromosomes from flaking and breaking. Now, you've got a new cell, 2 new cells. The chromosomes must now function. The chromosomes must come alive and produce messenger RNA so that we can get protein synthesis going and get this cell rejuvenated. So now, with that tight chromosome or piece of rope, plastic, in your hand, twist it opposite the way you would have to tighten it. Forcibly untighten it and let it flake into a million fibers. You're exposing the chromosomal genes. You're exposing the DNA. And under the microscope, it no longer will look like a chromosome. It looks like chromatin, okay, all dispersed. When this cell gets ready to divide again, this will once again tighten up. Okay, chromatin down here, dispersed genetic material. The nucleolus, the darkest spot inside of the nucleus of the human being. This spot disappears during division, but we're not talking division right now. This nucleolus here is responsible for the formation of what we call transfer RNA. I'm not going to go into DNA replication. I'm not going to, that's not our job here in anatomy. It was fun when I was learning it, but it just has no place here. But to remind you that this is what goes on in the nucleolus. Transfer RNA is produced, and that transfer RNA, the messenger RNA, and the ribosomes you've heard about before are all necessary for protein synthesis. The nuclear membrane is a double membrane. It shows here as being thick. It's a double membrane. The plasma lemma we left behind a moment ago is a single membrane. Why is it a double membrane? Does it have to be? Maybe it doesn't have to be, but it is. And so we report in anatomy on what things are. The idea, though, keeps coming up that when cells were being formed originally, some were bigger than others, some phagocytized or destroyed others and used them as food. And this cell here would be an example of a cell that possibly engulfed a smaller cell, usually devouring it. But some small cell managed to survive. It was genetically different or predisposed and to survival, and it stayed inside. And it had now two membranes around it, its own and the one that happened when it entered into the cell. And now we have a symbiosis starting up with the nucleus and the rest of the cell, which might actually be, in some people's mind, two cells living together. Very successful anyway, because it's around today. It's around today in the eukaryotics. Don't worry about the word. Centrioles. Centrioles, are they shown here? Yes, right here. The centrioles are going to put out fibers called the spindle fiber that guide the movement of the chromosomes during division. This little piece, which is L-shaped, splits in half. Half of it ends up on this side, half on this side. I don't know how many books will tell you that it splits in half and one piece migrates to the other side. That's bullshit. Okay, it splits in half and the two pieces migrate away from each other until they're on opposite poles. Set up the fibers across, which we'll see a little bit later, not today, and set up the spindle apparatus for division. Now, I haven't shown you the spindle apparatus. I'm going to, but I'm going to have to assume that some of this is ringing a bell for you, or that chapter 2 is going to also help you out. Endoplasmic reticulum. Coming away from the nucleus, the double membrane comes out as tubular tunnels. Some of these tubular tunnels have ribosomes attached, little tiny ribosomes. That's for protein synthesis. The endoplasmic reticulum is known for protein synthesis. It then continues on into other tunnels, but these do not have the ribosomes. These are called smooth endoplasmic reticulum. The endoplasmic reticulum, endo, inside, plasmic, the cell, reticulum, a network or a maze. Okay, here's the ER, the endoplasmic reticulum, from the pointer right now through this rough area, which is the rough endoplasmic reticulum with ribosomes, and including the smooth endoplasmic reticulum down here. And here's our label for it. While the rough endoplasmic reticulum produces proteins and enzymes, and all enzymes are proteins, okay, the smooth endoplasmic reticulum only transports them, moves them, and it eventually will go to a body that looks like this. They're not showing the connection. This is the Golgi body or Golgi apparatus, named after a guy named Golgi in the 1700s, who looked at cells of some plants that had large cells, and he looked with the microscope that he made himself and used a drop of water for the lens, and looked up and down and drawing after drawing after drawing at different levels until he finally could put it together in his mind, a 3D picture of the Golgi apparatus. I think that guy deserves to have his name attached to that forever, but they're trying to take it away from him. They don't want to have anybody's personal name in anatomy anymore, it seems. At least there's a group of people who don't. Okay, the Golgi apparatus accumulates the protein that was produced by the rough endoplasmic reticulum. And most of this protein is proteolytic. That means it's protein that destroys other protein. And it's released in these small little vesicles here, 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 and over here. They get larger as they suck up some water from the cell. This is called the lysosome. The lysosome, sometimes known as the suicide packet, goes into the cell looking for anything that maybe shouldn't be there, like a bacterium, or is there, but it's not functioning because it's gotten old, and it destroys it, releasing the material into the cytoplasm, and then it can be utilized as an energy source by the mitochondria. Here, here, mitochondria, here, mitochondria. You know, they always make these things look like pickles or sausages. Mitochondria don't always look like that. If you took a pickle and you cut it across and looked at it end on, it'd look round, not oblong. Some of them are actually Y-shaped. It's okay. Just know this. The endoplasmic reticulum, a network of tubules. Okay. The rough portion of that, the RER, rough endoplasmic reticulum, site of protein synthesis. The smooth endoplasmic reticulum, site of packaging and translocating, moving. The Golgi apparatus itself, the area of lysosome formation and protein accumulation. Skip vacuole for a moment. The lysosome itself is the vessel containing the proteolytic enzymes. The mitochondrion, responsible for cellular respiration. If you know what the Krebs cycle or the TCA cycle is, that occurs in the mitochondria. If you don't know, or more likely don't remember, we're not going to stimulate your memory here again, okay? I'm not going to ask you about the Krebs cycle. I'm not going to ask you about the TCA cycle. I just wanted to put that in there. One thing I will ask you is, what is the site of cellular respiration? Okay, respiration isn't breathing. It's degrading food or chemicals to energy. The mitochondrion is the site of cellular respiration, responsible for cellular respiration.
Charlie,
Yes,
is the mitochondrion separate from the mitochondria?
mitochondria is the plural.
Gotcha. Thank you.
Do you mind repeating what respiration means in cells?
Respiration refers to degrading potential food particles or molecules
Okay,
and breaking it down and forming energy. The energy will be ATP and NADPH.
okay, thank you.
This word vacuole. Vacuole is sort of like a bubble inside the cell. Let's see, here's one here. Okay, this can be brought in from the cell membrane itself. You ever look at a golf ball? Sure you have. It's pitted all the way around. Do you know how many pits are on a golf ball? How many little dugout areas on the surface of a golf ball? I had a guy in the class once who knew everything about golf. He knew the number of those pits. I think he told me it was 258, and how fast
Is it 374?
274,
374.
374. Wow, I know it wasn't that high when he and I were talking about it, but they've improved the golf ball since. How do you know that?
My husband plays golf.
And he won't leave you alone? I mean, he has to tell you about things like that?
Right. My husband plays golf too, and I had no idea how many of those were on a golf ball.
374. Tell your husband. And don't tell him that you learned it from another student. Okay. Okay, you all seen those little dimples? I don't know what to call them. But in a cell, they're constantly sucking in. And they'll either suck in just fluid, or if there's something like a bacteria passing by, they suck that in. If they suck in just fluid, they're a pinocytic vesicle. And if they suck in something solid, they're a phagocytic vesicle. The lysosome attaches to the vesicle. And if it has a solid inside of it, like a broken red blood cell or something like that, it devours it. It breaks it down for the mitochondrion. Okay. And this process is called phagocytosis. Phagocytosis. If it's a pinocytic vesicle containing only fluid, the lysosome breaks it down and releases the fluid, probably in a more digested state. The process is called pinocytosis. They're the same vesicles, caused by the same engulfing, if I can call it that, from the cell membrane or plasma membrane. It's just, do they have solids inside or just liquid? If it's solids, they're going to be subject to phagocytosis. And if not, it's going to be pinocytosis. Pinocytosis here can take in fluids. It can also take in salts, if we consider that a part of the fluid. And that's what it's showing here, and releasing them to a pinocytic vesicle inside. In phagocytosis, it can also take in a larger structure, like a bacterium here, or possibly a broken or dead red blood cell. And it is brought inside the cell for the lysosome to digest but it's a phagocytic vesicle, also known as a food vacuole, and it will be broken down. This is phagocytosis bringing in food, pinocytosis bringing in fluid. The words translate into phagocytosis, cell eating, pinocytosis, cell drinking.
Charlie,
Yes?
for phagocytosis, would it also include the fluid, or is it only solid material?
No, no, no. The fluid is always there.
Okay.
The fluid's always going to be there. Okay. Let me get rid of this. And I'm going to give you a break. And when we come back, we'll do those phones that we have to do. It'll take a break till 2:20. Okay, so you better hop on it.
Is his internet cut off?
No, it's just we're taking a break.
Oh, okay.
Okay. I only give us 10 or 11 minutes. Is that enough for you guys, or do you want more? Or you want less, or do you want, you know?
It's
10
fine
is
have
with
fine.
me.
Thank you.
Huh?
It's
10 is
fine.
perfect.
I think we're talking over each other.
And
It's
it's
good.
good.
Mhm.
Okay, yeah, it depends on what you got to get done. Basically, I only refill water, so 10 minutes is fine with me. But if you need more as a group, just sort of let me know. Otherwise, we just get going and finish our material for today. Any questions that you came up with during the break?
How's your day?
This has been my day. You have shared my day. This is it. I was up till about 4 last night, so I essentially get up maybe 2.5, 3 hours before this class. Uh, that's it, and here you are. And I spent the morning looking at the emails and trying to get the distribution list together, that kind of stuff. Real boring, real boring. Okay, this is much more fun. This to me, this wakes me up. I don't know you. When I was a student as I said, UCI, I was pretty much bored all the time, and mainly because the instructors at UCI, in the beginning, I'm not talking about today. I don't know about today. Uh, they did not want to teach. Most of them did not want to teach. They would send in, say, from the physics department, they would take one guy, one professor, and send that one out to teach all of the physics lecture classes. They had TAs, but they did it all. Nobody wanted to do that. But by doing that, then all the other instructors could do their research and never had to see a student at all. And then the next quarter, because it was on the quarter system, the next quarter, somebody else had to do all the lectures. And then they kept rotating it because they did not want to be with the students. And it showed. It showed so badly. We had to call him Doctor too. Shaw was his name. Uh, he came out and he got so bad, so disturbingly uh, boring that people started bringing in tape recorders. Okay, in those days, tape recorders were about the size of a pack of cigarettes. Okay, and we put the tape recorder up on the front desk, the demonstration table. Uh, Physicists didn't do any demonstrations, and he would talk and mumble and try to remember what his name was. And unfortunately, uh, we would go up and get our recorders. People would take them with them. Sometimes you put two recorders, one for you and one for your friend. But somebody put a tape recorder up there with an explosive in it, and it went off right in his face. Not a deadly explosive, fortunately, but enough to scare the hell out of him. And after that, no more tape recorders. And so that meant everybody had to really be in class listening to this drivel. It was awful. I actually had an 8 o'clock class the first Monday that the school actually opened. I had a 9 o'clock class and a 10 o'clock class, and they were all in the same room, the science lecture hall. So I just stayed in the same chair for 3 hours. The second person after the physicist was the social science. Oh, social science. That's the most boring subject I've ever come across. At least they made it that way. But then we had one good instructor. One good instructor. He was chemist, and he was teaching us just general chemistry. And it was a step down day for him. This guy's name, you wouldn't think. His name was Sherry. This is a guy, a chemistry instructor, big, burly arms, looked like a football coach instead. And his name's Sherry. Sherry Rollins. And if you knew his real name, it was Sherwood Rollins. And he won the Nobel Prize just a couple years ago. So I had a really good teacher there. Once in a while. Okay. Don't ask me questions like that. Anything else? Okay. We'll go back to the share screen. And yes, I am recording still. And can you get that to the right size? Back up. Okay. You see the cursor in the middle?
Yes.
Okay. We really did find a good shortcut yesterday for me. I probably should have known it. Okay, what I'm going to do is tell you first off for today, 610, okay, the bones and figure locations is always going to be helpful. It's especially helpful even when you're on your own studying. I've copied it for myself. Here's what we're going to look at today. Upper appendicular skeleton, the 126 bones of the upper of the appendicular skeleton. That would be 126 upper and lower. And it has figure numbers that correspond to the figures in the book that we're going to use. If you didn't print this out for yourself, that's okay. You'll have it later. But you'll see what we're going to be using figure 8.2 to begin with. And then backing off to 8.1 and figure 8.3, 4, not 5. I don't know what 5 is, and 6. So we'll be using figures 8.1 through 6, but not 8.5. Okay. This should help you follow along if you have it. If you didn't have it, you'll get it later. So that goes along with this last topic here. Now, over the weekend, probably tomorrow night about, I'm going to be inserting another quiz. Okay. The quizzes are for you. Okay. You might be interested in this. I don't know. I gave you a quiz and said take it after the second meeting. We're at the fourth meeting now. How many people do you think have taken the quiz?
80%.
Okay, there's 36 people in the class. So 80%. 25. Okay. I don't think so. I haven't looked. Anybody else want to make a guess?
12.
Okay. I think you're probably closer. Let's find out. 16 people. Okay, not quite half the class has taken the quiz. Okay, their average score, 88%. Well, that doesn't mean that they only took it one time. Some may have taken it 2 times. Here are the questions. Here's the ones that people got correct. And the black ones tell you how many people missed by choosing that question. So you can learn a lot from these quizzes. Unfortunately, you don't get to see that page. If you ask me, I'll certainly show it to you. Okay, now modules. Okay, upper appendicular. This is one of the easier sections, I think. This is scapula, and you're looking at it from anterior view. You're looking at it from anatomical view. Okay, this is if you were looking at somebody and could see right through them. Okay. At the very top of the scapula.
I think, right.
Okay, put it on mute. Okay. Here's the superior angle. We have roughly a triangle. Pretty nice triangle, actually. The superior border is across the top. Okay, this is the superior border here. The arrow or pointer is touching the bone, but the superior border is really all the way across the top. Down the spine, your spine is located here. This is the medial border. It's also known as the vertebral border. If you want to use that term, you may. But medial makes a lot of sense. And on this side, underneath the arm, is the lateral border. It's also known as the axillary border, and you may use that term. What's axillary mean? It means armpit. Okay. But lateral, superior, medial borders. At the top, we have the superior angle, and at the bottom, the inferior angle. Now, many of you can, you get a niche back or whatever, you can reach most parts of the scapula. But if we haven't stretched in, say, months or years, or body shape is just such, then we can't always reach everything. That's when somebody wants a back scratcher or a friend. Okay. This little process here coming out at you and curving over, to me, it looks like a broken little finger. This is the coracoid process. It's going to be a site of attachment for the biceps brachii muscle of your arm. What I say isn't important yet. It will be when we get to the muscles. But what I point out and say also that this is the coracoid process. That is important. That's one point on the test. It'll be there. In the background is a large protuberance called the acromion or acromion process. When we turn this around, you'll see it better. The lateral angle is not important. It is not on your study guide. As to the study guide itself, that was the list that I showed you a moment ago with the figures, and some of you have it right in front of you right now. Others can imagine it though. That list of material is the list that is picked from for making the exam. If it's not there, it cannot be asked. Lateral angle is not there. I can't ask it. If I should mistakenly ask something that I previously told you it's not there, well, then I give everybody credit for it. You always hope for me to make a mistake like that. Okay, you're looking at the scapula again. In this area in here, which looks like a trowel. Here's the handle, hold it, and you can scoop sand with this. This is the subscapular fossa. Subscapular fossa. The word body does not appear on your study guide, so we don't need to worry about that one. Okay, if you feel that that's not enough time, well, the picture is always going to be here. Plus, we're going to look at the other side now. Both sides, in fact. Here's the side on the left that we just looked at. This is a right scapula, however. Fits in right here. Subscapular fossa, superior angle, medial border, inferior angle, lateral border. Don't worry about these. Coracoid process, an acromion or acromion process, and the superior border across the top. Now, turn it around. We're looking at somebody from the back. We've come up behind them. Okay? Slap somebody on the back or pat somebody on the back. Here's the scapula from the posterior side. It has a spine right here. This is called the scapular spine. You can just call it the spine. It's not the same, of course, as the spine of the spinal cord. The spine terminates in this large fan-like piece of bone. This is the acromion. This is also known as the acromion process. And you'll see in a moment how the acromion helps to hold the head of the humerus in place, the top of the humerus. Put your mind with my cursor here on the spine and go up above it. There's a trough. There's a trough. I often take a marker pen and put it in that trough. Well, I'll be able to do that with you on Monday when I get the bone box out. And the marker will lie in here. That trough is the supraspinous fossa. Now, if you're thinking a fossa is like the mandibular fossa, nice and round or oval, no. A fossa is a depression, and it can be large like this. This is the supraspinous fossa. What fits in there nicely, snugly, is going to be the supraspinous muscle. Obviously, you don't need to know that now. That's in the second unit. Below the spine, this whole area here is the infraspinous fossa. Now, don't be confused just because it's not labeled. This is still the medial border here and the axillary or lateral border over here. Okay, now we've got one more addition. We've got the anatomical view over here on the left. We've got the posterior view that we just looked at over on the right. And now we've got a lateral view. The arm would dangle from here and in front of it. Okay. This plate, which is about the shape and feel of your thumb print. If you put your thumb on that, it would cover it perfectly. That's the glenoid cavity, sometimes called the glenoid fossa. But glenoid will always be in the term. Now, if you look at this glenoid cavity, and here is the acromion process. Now, here, this one calls it the process. And over here, that bent pinky finger, the coracoid process. The rounded head of the humerus can fit into here. And with ligaments stretching from the humerus to these 3 points, this one, and the back of the glenoid cavity, we hope that the humerus is attached. Actually, the greatest amount of attachment of the rotator cuff, which is the shoulder, is the muscles that we don't see here yet. But you probably also know that the arm or shoulder is subject to dislocation, as are other parts of the body. And the attachment is not perfect. Okay, I'm looking at this, and here's the lateral border. Now, the lateral border is over here. When we turn it sideways, you see how thin it is. Here's the lateral border, glenoid cavity. Back here's the spine coming up to the acromion, the bent finger, the coracoid process. This backs us up to a figure. I don't know the number myself now. Is it figure 6-1?
8-1.
8-1, of course. Okay. This is the clavicle. Clavicle does not have a lot of parts. Clavicle is the collarbone. We'll never use the word collarbone, though, on an exam. This is the clavicle. The clavicle is the most frequently broken bone in the body. If I were to ask you, have you ever had a broken bone? And those of you who have answer, you'll say probably it's the humerus or something, but not necessarily the clavicle. Why is the clavicle the most broken bone then, when it's not in your experience? Well, it's because as people get older, they get a little bit less steady. And when they fall, like anybody else who's falling forward, you put your arms out in front of you. And the pressure on the clavicle of a person falling like that is what causes it to break, especially in an older person when it's already probably got some degeneration to begin with. So when it comes to just adding up the shear totals, the collarbone is the winner. Okay. One end, let's just look at the top one. One end of the clavicle is flat. It's like if I took the end of a roll of clay or a rod of clay and, with my thumb and index finger, I squished it. This is the acromion or acromial end, I should say. It's not the acromion. The acromion is only on the scapula. This is the acromial end. It's going to articulate with the acromion. The other end is chubbier, thicker, and it's the sternal end. The sternal end goes into the notches in the top of the manubrium that we call the clavicular notches. We saw those yesterday. Okay. So if you're looking at this guy here, the sternum would be here, and this would move away till you got to the shoulder. At the bottom, there's a bump. When you go to the acromial end, the flatter end, pull back a little bit, and there's a bump. Up here, here's that same bump. This is the conoid tubercle. A lot of C words today, words starting with C. Conoid tubercle. It sits down on top of the first rib to give a little bit of stability. Okay. That's it. You don't have to worry about anterior, posterior. You don't have to worry about the body. You just need the two ends, the acromial end and the sternal end, and the conoid tubercle. Okay, well, we're leaving it behind. Here's the scapula and the clavicle here. We need to attach to it the humerus, the arm bone. When we say arm, we're meaning upper arm. If we want to say the lower bones of the arm, then we've got to say, hopefully, that we're talking about the forearm. Okay. First off, one of these is anterior view, and one is a posterior view. Let's look at it and figure out which one it's going to be. Let me magnify it a bit. Okay. Here's the head of the humerus. I think we can all agree with that. Looks like a skull cap. Underneath the skull cap, follow my pointer, going up, or on the other side, going up, is the anatomical neck. Okay. It's not small as it was on the rib yesterday. The anatomical neck goes all the way around the base of the skull cap. And then roughly here, a couple inches down from the head, if you were to hold it with your fingers, your hand, and use your index and middle finger as a pair of scissors, if you were to scissor it right at that point, well, you'd understand why they call it the surgical neck. That's where it breaks. That's where it breaks. The surgical neck is not an anatomical feature. It's just a medical description. Now, which one is facing us and which is not? Look down at the distal end. Here, we've got a small little fossa. Over here, we've got a large fossa. The large fossa is there to fit with the ulna of the forearm and make the elbow. I feel like my elbow will crack today. No, it's not there, that one did, but you didn't hear it. Okay. If you find your elbow on the back side, you have found this hole, which we'll name in a moment. But this is the posterior side. We need to turn it this way. Okay. Or direct your view this way. Okay, now start in the middle, go to the head, past the anatomical neck, and all the way across to the outside here to this structure. This structure or bump is the greater tubercle, cross a small chasm coming back, and we have the lesser tubercle. The tubercles are for the attachment also of the bones of the muscles of the humerus. Okay. The greater tubercle is larger. You have to see it from the side. It's here, over here. The lesser tubercle is right at you, points right at you. The greater tubercle points laterally. The groove in between is the interturbicular sulcus. Here they're calling it interturbicular groove. Groove and sulcus are the same thing. I wish they'd standardize it too. Each book seems to go its own way. Interturbicular is the key portion. Now come halfway down the anterior side of the bone and the lateral portion over here. There's a bump. This is the deltoid tuberosity. The deltoid tuberosity is the point of attachment of the deltoid muscle. There's 3 parts to the deltoid muscle that you'll learn later, and all 3 of them attach here. And they need something to grab onto with a roughened surface, and that's the deltoid tuberosity. Come all the way down now, and to the 2 points. This one lateral, because it's on the lateral side, and this one medial, because it's on the side that the head is on. This is the medial epicondyle. This is the lateral epicondyle. I'll show it to you from the bone box on Monday, but if you can, I'm doing it. I know you can't see me. I just want to say it right. Take one hand and go to the other and grab your elbow from the bottom. Grab your elbow from the bottom, spread your thumb and index finger apart, and find the 2 bumps, one on the lateral side, one on the medial side. That one on the medial side you used to call your funny bone. If you can't do that, don't worry. I'm having you look for these so that you can feel them. They probably feel like they're quite far apart, but you have a lot of connective tissue associated with them. And when that's gone or is ever removed, then it'll be smaller like you see here. The lateral epicondyle and medial epicondyle. Come down below, and on the lateral side, we need to find the capitulum. You're trying to find it with me on a piece of paper. The capitulum is rounded, and then we have a trough. It's called the trochlea. I'm not liking this picture.
Charlie,
Yeah,
the capitulum is on the one side and the trochlea is on the back side, correct?
no, they're both
Or
on the
no?
front.
Okay.
Let me,
Maybe it's just the picture then.
that's why I'm changing pictures. I don't like it now myself. And I hope I do this right. Okay, here's the bone that we were just looking at. Okay, and it's probably got more things that we need. No, it's actually got them all. I can't you come down on the bone on the left side of the diagram. Okay, in other words, over here. And I'm coming down. And I'm going to find this capitulum. Here's the capitulum. Capit means head. So this is supposed to look like somebody's head or an old statue that's been weathered away. This is the capitulum. The head of the radius in the forearm revolves around it and will swing up and be stopped as you raise your, you flex your forearm. Bring it up. You can't bring it up all the way. You get stopped when the head of the radius goes into this pocket right here. This pocket is the not labeled here, it's labeled in your diagram, is the radial fossa. Radial fossa. Here's the capitulum. Here's the radial fossa. Here's the lateral epicondyle. And the more pointed one, the thicker, bigger one, is the medial epicondyle. This is the trochlea. And the ulna, which you haven't seen yet, of the forearm rotates up and stops in here, in this pocket, the coronoid fossa. So if I go back to my last picture, here's the coronoid fossa right here. Here's the trochlea, the capitulum, the lateral, the medial epicondyle. Only thing not shown in this picture or labeled in this picture is the radial fossa right here. That's the easy one. If you look at the capitulum and the trochlea, turn your head sideways to the tilted left, so that this is at the top. This may look like a pawn in a chess set to you. Capitulum, trochlea. Don't be afraid at all of Googling new pictures for yourself. They really, really can help. Even if the picture that I have posted is the best one for the entire class, it's not necessarily the best one for you. Okay, so now I'll go back here. And we find that we have labeled everything on this side. On the posterior side, the greater tubercle now turns over to this side, no big deal. Come all the way down. The medial epicondyle now flips to this side. And here's this large cavity. This is the olecranon or olecranon fossa. I'm trying to change from olecranon to olecranon because I've asked many, many people who also never heard it before, instructors, how they pronounce it in class. They're going with olecranon, and that's not bad. Okay, this is the olecranon fossa, and the head of the ulna fits in there. This is a little quiz right in the middle of things. Sometimes these quizzes are great, and sometimes they make you furious. So I always put in the answers if it's a quiz right away. the star. The star is on. You want me to say it or you want to try it?
Is it the lesser tubercle?
That's exactly right. That's the lesser tubercle. Making this?
Greater tubercle,
Yes, it is. What's this structure up here?
the head.
The head.
Yes,
The head.
the head. Yes. And these are trying to show the anatomical and surgical neck. I would put the surgical neck down here myself. There's no structure that I've given you other than anatomical neck that would fit this one. We'll see what they think it is in a moment. Here, look at your notes.
Deltoid tuberosity.
Deltoid tuberosity. This one, the rounded head-like structure.
Capitulum.
Capitulum. And the depression above the capitulum is the radial.
Fossa.
Which one?
Coronoid fossa.
It's the radial fossa.
Radio.
Oh.
What's this structure here that looks like a spool of thread?
Trochlea.
Trochlea, right. And the cavity above it, the star shows better than the cavity. This
Coronoid?
Yes, coronoid fossa. I always remember it. You know, I wish they'd called it the ulnar fossa. They did nice over here calling this the radial fossa because the radius is going into it. Why not call that the ulnar fossa? They didn't, and we don't get to. So it's the fossa in the arm that's closer to the heart. Remember how I did that with the mandible? Okay, so this is coronoid fossa.
Charlie,
This is, yes?
radial and the olecranon fossa are same thing?
No. No. The olecranon fossa is on the other side. Okay, can't show it to you right here. Here's the medial epicondyle. And here are the presumed answers. Look, that's what happened. See, there were 4 lines here. There's only 3 here. Somebody took out one of them because it didn't work. You know who it was? It was me. Okay. But these are good. To look at the olecranon fossa, it's right here. It's very large. And on the posterior side.
Charlie, I have a question.
Yes?
In the picture with the black background there, how would you be able to tell if it's a posterior view or an anterior view?
The anterior view doesn't have the olecranon fossa. The posterior view always has the olecranon fossa. And, you know, you're right, it doesn't really show so well.
I guess because I was getting it a little bit mixed up, the coronoid fossa and the olecranon fossa.
Let me solve it.
Okay.
Okay, here's the olecranon fossa on the posterior side. See how big it is?
Uh-huh.
And then on the anterior side, not the case. Here's the trochlea, but here's the olecranon fossa, large.
Mm-hmm.
I'm still not happy with it. So give me a moment. That's okay. Here's the ulna, and it goes into the olecranon fossa from the posterior side. You can feel it on yourself if you feel the back of your elbow. Find that pointed part of the elbow. You might, when you're very aggressive or not necessarily hit somebody, with your elbow, and then go up your arm. Okay, and that's where it is. Here's a good picture. That's the olecranon fossa. One fossa. On the other side, there's a radial and a coronoid. Okay. This is the ulna. This bone looks either like a snake, it looks like a carnivorous plant, it looks like a wrench. Whatever you see in it, if that helps, fine. Whoa, oh, what's she doing here? Okay, the very top of the ulna, there's a process, a large process. This fits into the olecranon fossa. This is the olecranon process. Sometimes it's just called the olecranon. Olecranon. That fits into the back of your elbow. It fits into the olecranon fossa.
Charlie,
Yeah,
on the test, if we were to put olecranon process or olecranon, would we get points either way? Or I guess it's multiple choice, huh?
it's multiple choice. So I won't confuse you by giving you two similar answers like that. And you see the root word, the olecranon, if you know that's what it is, then you're home free. It's multiple choice. Okay, now you look at this, the mouth, I call it. Okay. This is the part that goes around the base of the humerus. This is the trochlear notch, sometimes called the semilunar notch because it looks like a moon. This is the semilunar notch. If you look at it over here, it's coming out towards you. Bring your imaginary thumb out and touch this little peak right there. Touch it right there. That's the coronoid process. The coronoid process fits into the coronoid fossa. When you flex your arm up, and this whole thing goes up, swings up as you're flexing your arm, this goes into the coronoid fossa of the humerus. This is the coronoid process. In an X-ray, which again we have yet to see, but we are going to, you find that coronoid process, and then you can find anything, anything. It's the key to the map. If I put my thumb on the coronoid fossa process and hold the ulna down here with the rest of my hand, just stick up your, make a fist, put one thumb straight up. Okay, now wrap the fist around the shaft and put the thumb on the coronoid process. Now, if I now go either right or left, this way or this way with my thumb is going to feel very comfortable and find a spot right on the side. That's where the radius is going to articulate with the ulna. Okay, and that'll be the radial notch. It'll be a syndesmosis. I know you're not ready for that term yet. We had it 2 days ago, though. Got to get thinking about it. The proximal end of the radius and ulna are joined by a type of suture called the syndesmosis, fibrous semi-movable joint. So we look for this spot here, and it's the radial notch. Okay. Come down to the bottom part of the ulna. Tap it to the tabletop, and only one point touches first, right there or right there. And that's the styloid process of the ulna. Styloid process. Styloid means like a feather. It means like a quill pen, an old pen that we see in cartoons and nowhere else. Okay. When you think about it, and I'm just thinking about it for the first time, what's the ulna got to do? What's the ulna got to do? It has to bend, so it's got to have this situation. And it's got to extend the arm, so it's got this length. That's it. Here's the radius, one of my favorites. The radius in this is upright position. Here's the head. This fits into the radial notch on the side of the ulna that we just saw. Head, neck, and then the bump is tuberosity. Remember, in the ribs, it's head, neck, tubercle. But in the radius, it's head, neck, radial tuberosity. And you get it wrong if you answer tubercle, and they are likely to be together in the same multiple choice question. Radial tuberosity, radial tubercle. Tubercle is much smaller.
The radius fits into, I'm sorry. Yeah, the head of the radius fits into which part of the ulna you mentioned?
Right there. The radial notch.
Gotcha. Okay, thank you.
Okay. It also revolves around the base here. It revolves around the capitulum. The capitulum stands right on top of the radius. And when the radius comes up as you flex your hand, it'll go around the capitulum and be stopped by the radial fossa, which is here and here with the label. At the bottom end, the distal end, to be correct, tap it on the ground or tap it on the table. And there's a styloid process here too. This is the styloid process of the radius. Your test would just say styloid process. And on the other side, okay, from the styloid process, on the other side is a notch, the ulnar notch. It's never very distinct. What fits in the ulnar notch is the ulna, the other end of the ulna, and another syndesmosis, because we use this as one of our examples of a fibrous joint that is slightly movable. Okay, so these two run side by side and bring us to the hand. I don't know how many times people have told me they expected the hand to be the hardest part. Sometimes your expectations make it come true, and it's not. The hand is one of the easiest things. Yes, it's got some memorization, but sometimes it just makes sense. Okay, so the hand includes everything that you see here. Okay. And these are your digits 1 through 5. Digit 1 is the thumb and is always lateral. 2, 3, 4, and then 5 is your little finger and is always medial in anatomical position. The digits that, themselves are composed of bones called phalanges. Okay, the singular of phalanges is a phalanx. Here's the plural over here, and here's the singular over here. The thumb has only 2 phalanges, the shorter one. It has the distal phalange and a proximal phalange, or phalanx. That's it. All of the distal terminal ending phalanges on the hand also have the fingernails. Your fingernail is on, in each case, the distal phalanges. Okay, that's an easy way to remember that one. Okay, let's go to digit number 2. Here's the distal phalange, way up here, small, because it's bent away from us. The next one back is called the middle phalanx. And then we have the proximal. There's always a proximal phalanx. There's always a distal phalanx. But on the 4 digits, number 2, 3, 4, and 5, there's also a middle phalanx. How many phalanges in one hand? 14. 3, 6, 9, 12, and then the 2 for the thumb, 14. You got 2 hands, you got 28 of these phalanges. So we've learned 28 bones in a couple of seconds with a little review. When we get to the feet next Monday, we're going to have the same number. So the 28 and 28 are going to be 56 bones that we learn in no time at all. The palm of your hand are these larger bones. These are called metacarpals. Metacarpals. Here they are over here. Metacarpal associated with the first digit is metacarpal 1. Metacarpal 2, using Roman numerals, metacarpal 3, 4, and 5. Metacarpal 1 is always lateral. Metacarpal 5 is always the most medial. Since there are 5 of them and 2 hands, you just learned 10 more bones. Okay, at least the names of them. And there'll be 20 more when we include the feet next week. So we're racing now. And here are the carpals. This is a plastic skeleton, you can tell that. Okay. Let me sneak a peek down here. Okay. And I have another set of bones. These are real bones, although they have been strung together. Okay. Let me use this one. Distal phalanx, middle phalanx, proximal phalanx, metacarpal 3. 1, 2, 3. Here's how you learn the bones in the wrist. And there's where people go, I can't do that. Huh, yes, you can. It's simple. Okay. Start with the thumb. Come down to metacarpal 1. Okay, that was easy. And notice that in the wrist, it articulates with only one of the 8 bones that are in the wrist. Now, the one thing you have to do, of course, is to learn the name of that bone. And that is the trapezium. It's labeled T2. I don't know what the 2 bit is about, but here it is the trapezium. Okay, trapezium. Go over to metacarpal 2. Come down. And it primarily latches onto and articulates with one bone, and that is the trapezoid. Trapezium followed by trapezoid. How are you going to keep those apart? You got the words trapezium, you got the trapezoid, that's fine. But how do you know which one it goes to? You're going to mix them up. They sound so similar. If you have to slow down, you can't, and you have trouble with it, do it alphabetically. Trapezium first, trapezoid comes next. Okay. Go to metacarpal 3, come down, it articulates as the only bone articulating with this large bone in the palm of the wrist of the hand. And this is the capitate, because again, it's supposed to have the qualities of a statuary face. This is the capitate. Nobody misses the capitate. Now go to metacarpals 4 and 5. 4 and 5 come down, and they both are fighting over this bone labeled H. That is the hamate bone. So the trick, it's really simple. Go to the metacarpals, come down, have it memorized what you're going to find down there. The hamate also has what we call a ham hock appearance on the other side. It has a bony projection. I can show you that picture, but you'll always be able to identify this because the 2 bones, 4 and 5 metacarpals, attach and share the hamate. There are 8 bones in the wrist, and we just did 4. And the 4 that we did all touch the metacarpals. The other 4 do not touch the metacarpals. They're down here under. So we go to the one that's most lateral. That'll be underneath the trapezium. And here's the bun bone. We've already done that. We've done that. We've done that. We've got to get this one tucked here. This is the scaphoid. The scaphoid bone. It's also known as the navicular. The scaphoid bone looks like a little boat. Now you don't see it, but if you take these and separate all of these little wrist bones, or carpals as they're called, you will see these shapes. This is the scaphoid. Now we're going underneath the capitulum. We've already done the capitulum, so we can't be going there. We're going to this one. This is the lunate. And believe me, it really does look like a moon. Where? Well, take it out, and it has a crescent shape. And the capitate is fitting into the bowl of the moon. Okay, well, let's go on one more. There's no other way to go but here. We've already done that. We've already done that. We've got to do this one. And this, unfortunately, has 3 names. Triquetral. That's what the page here has it as. Triangular. I like that one. And triquetrum. Attached to that, there's one bone left. And that bone is right here. The shape of a P. the pisiform bone, pisiform.
Is that the one that you can only see on the interior view, but not on the posterior view? Is that how that one works?
Usually that's correct. Usually it's correct. But once in a while I've seen pictures where they somehow have somebody that it's different in. It's the roundest one that you'll ever spot, and it's up there next to the hamate. Over here on the plastic model, it's going the other direction. Okay. Here's the trapezium. It's bent back. From number 2, this is the trapezoid. From number 3, the capitate. And from 4 and 5, the hamate. Coming back to the next row, this is the scaphoid or navicular. This is the lunate. This is the triangular in here. And you can barely see where they separate right here. This is the pisiform. Okay, any questions? We can go through them pretty fast sometimes, but maybe sometimes we should slow down and sort of review them after you've had a chance to study them and look at them. The tougher of the two, a practicum or a lecture test, is always the lecture test. Okay. Well, why? One's conceptual, the other's visual. I think so. Okay, but I'm just asking you not to neglect one over the other. You have first the practicum coming up. That would be first and foremost in my mind, of course, but don't let the other slip, no matter sometimes how easy you think some of the material might be. Okay, any questions? Okay, then I'm going, yes.
In reference to the PowerPoints and the figures in the textbook, is one picture over the other better to study or just study and know them both?
It's not quite sure. Where are you getting, what PowerPoints are you talking about?
The slides that you just went over,
Yes.
those slides, or the pictures in the textbook, the figure 8186. Is it better to study?
When you're looking at figures, okay, that really is your personal choice. Which one helps you identify everything? And then make notes. You weren't able to find this in either of the ones, and email me, and I'll find it for you. Okay, or if you think it's won't take forever and a day, or well, yeah, just bring it to class and ask it the first or at the end, like we're doing right now. You know, I'm not going anywhere until the last person is gone in a moment. And if you've got questions that you've accumulated, I'm there, and you can ask me. You know, until our timer runs out, and then I can start a quick launch, and we can continue. Okay, but we will get it done. So those of you that are ready to exit, be my guest, and thank you for coming, and have a good weekend. And do look over your mail for some material from me. I always want to punish somebody. Okay. And I'll wait here and answer questions for those that may still have them. Okay.
Charlie,
Yes.
first I want to mention the skull underneath your Christmas decor behind you.
Oh yeah. I got a lot of skulls in here.
I just noticed that it's like a Christmas horror movie or something. Quick question though, speaking of the practicum tests next week, I think our discussion for next week has to do with us formulating a question that is similar to one that could be on a practicum exam. And it
Yes.
said you would show us a few questions as examples. Is that something we're doing on Monday, or are you not doing that?
No, I'll do it on Monday.
Okay.
I'll do it because I'm not worried that you need to do it over the weekend anyway. You've got plenty of other stuff to keep you busy. I did have the last 3 classes, 2 classes, devised questions for a practical exam. I didn't use their questions, but the exercise I think proved very helpful to them. Made them think, and they did an incredibly good job, really good job. Okay, so I'll be posting the second week material over the weekend. Okay, I like to get the entire week's worth of material all up and posted, and then I can relax.
Charlie?
Yes.
So for next week, for our discussion thing that we have, is it going to be focused on the material that we're studying right now, or is it some separate?
It's usually separate.
Okay.
Yeah, I'm not saying it will be. It usually what you know. I am not going to get into whether we should wear masks or not. We'll just get an argument and find out who's, well, you know what I mean. But I do have questions about your careers and how to best aim for being successful. I used to have questions on vaccination, but almost everybody in the class went for it and said they're getting it, they're getting it, they're getting it. And maybe you guys are not the same. But anyway, it's medically related questions. Okay. And they're not meant to be challenging. I just want your opinion. I want you to share your opinion, actually, with the other people.
See you, Charlie.
Bye-bye.
Bye, thank you.
Bye.
Bye. Bye, everyone. Thanks, Charlie.
Take it
Thank
easy now.
you.
Bye, thank you.
Hi, I had a
Yes.
sorry, I had a question still regarding the outline. So I have already outlined chapter 1 and 9, and so the ones that we have to turn in for Sunday is 1, 9, and 6.
Okay, first off, you don't have to turn it in for Sunday.
Oh,
Okay. I talked about that earlier today. Maybe you weren't there at the very beginning. Sunday's the recommended day. Why? So that you can get it out of the way and get busy, with your other stuff. But you have until the morning of the practical exam, which is on the 17th. Okay. They have to be turned in before the practicum. That's all. The other date, I took it out. Take a look. Okay.
okay. And then I could just check on the modules to see what chapters have been assigned for the outline.
It's 1, 9, and 6 if you wish, or 1, 9, and 2 if you wish. Okay.
Okay, thank you so much for the clarification.
Yeah, I changed my mind in the middle of things, and I don't want you to be inconvenienced. If you've already started 2, then include 2. If you barely started 2, I'd set it aside and pick it up again when we're ready for 2, and do 1, 9, and 6. But it's your choice. 1, 9, and 6, 1, 9, and 2.
Okay, thank you.
Okay, thank you, Jennifer.
Thank you, Charlie. Have a good
Okay.
weekend.
It's you 2 again. Okay.